Okay, everyone, rounding out Unit 3 is going to be Chapter 7, Respiration. So we have discussed in this unit so far generally energy and what that looks like when we build up and break down reactions. We've also looked at photosynthesis, which is how food gets generated. And now we are going to look at the process of where we take that food and can actually break it down into some form of usable energy, which is respiration. So photosynthesis is considered anabolic, meaning that you're building food molecules or more complex molecules from simpler, smaller ones. Respiration is really the opposite of that. It is catabolic. It is breaking things down. So it's going to essentially break it down to extract energy from those molecules, like glucose. And you can see at the bottom is the overall general equation for cellular respiration. So glucose's formula is C6H12O6, oxygen, and then which we need to breathe in order to do efficient cellular respiration. And then we can breathe out CO2, generate water, and harvest a whole lot of energy, which is not shown in that equation. This just kind of highlights what that means. So you have a complex organic molecule like glucose, and then the catabolic process of actually breaking it down. We have simpler products. Not all of them are waste products, but some of them are going to be used to facilitate other reactions later on. And there's a lot of heat that's given off in this process too. Generally speaking, in an open system in which, humans and other organisms are open systems, cells require energy to perform work. So nothing happens for free. You need energy to do chemical work, transport work, mechanical work, and so on. So this graphic just kind of puts it all into perspective. So energy flows into the system as sunlight. So we're open because we are receiving sunlight. And then those autotrophs, which are organisms that can make their own food, basically do photosynthesis. They can transform that chemical energy into organic molecules. And in that process, they release oxygen, which is awesome because we need that to breathe and perform cellular respiration. So cells use some of that organic molecular chemical energy and then can break it down to make ATP, which is kind of our cellular form of energy. In that process, we oftentimes give off heat. We also give off carbon dioxide and water, which is great because then the plants need those things again to make more food. So that's why it really is kind of an energy cycle in this process. Here's kind of the two equations just back at you, seeing how they line up with each other. As you can see, they're basically just the reverse. So photosynthesis is an endergonic reaction. It is endergonic, meaning you have to put in energy to make it happen. In this particular case, light is the energy that is making that happen. It's endergonic. It's also building up molecules, so you could say it's anabolic. It's got a positive Gibbs energy change. There's a lot of different things that you could say about it. If you take that equation and you just reverse it, it is cellular respiration. So this is exergonic, meaning that it's giving off energy or has a negative Gibbs constant. These are the spontaneous ones because they don't require the input of energy. And you can see that it's being broken down. So it's a catabolic process. And in the overall reaction, we generate ATP. Back to chemistry. This particular slide is highlighting how cellular respiration itself can really be a redox reaction. So redox is short for reduction and oxidation reactions. Oxidation means that you are losing electrons, oil. Reduction means you're gaining electrons, rig. I've always remembered it as oil rig, but if you're one of those people firmly in the camp of Leo Ger, where losing electrons means oxidation and gaining electrons means reduction, go for it. But really, I think the best way to think about this is pretending that the hydrogens in these equations are electrons, because they really are. Hydrogens are composed of one proton and one electron. So when things are gaining electrons, they are being reduced, but in our overall equation, it means like it's gaining an H, which is where the electron is flowing with in this case. So anything that doesn't have an H and then has an H is going to be reduced. So in the bottom, for example, that's oxygen gains hydrogens, also electrons. So it is being reduced to water, whereas glucose is losing those hydrogens, electrons, and therefore it's becoming oxidized into carbon dioxide. This is another graphic showing you pretty much that exact same thing, just with a non-glucose example. So you can kind of see overall in the structure what's happening to the electrons and where they're moving. You can see that they're moving along with the hydrogens. So if it's gaining hydrogens, then it is being oxidized if it's gaining hydrogens, it is being reduced. Oil rig, gaining electrons is reduction. Okay, so energy harvest. We talked about this a little bit in photosynthesis, where in the photosystems, the electrons have to kind of fall down an electron transport chain. This is going to happen in here too, for the same purpose. If you have excited electrons dropping down to a stable electron state, they're basically going to explode off all of their energy. And the cell can't really harvest all that energy in a quick fashion. So it drops it down slowly and can then harvest that given off electron energy. So that's why it says the energy is released as it falls from organic molecules to the electron acceptor at the very, very bottom. In this case, oxygen is the final electron acceptor in cellular respiration. The breakdown of steps means that it's going to go from food to NADH, which is one of those shuttle carrier molecules of the electrons. Then it's going to fall down the electron transport chain to the final electron acceptor, oxygen. The coenzyme NAD, which we saw in photosynthesis, similarly was NADP+. NAD is another electron acceptor here. It can pick up 2 electrons and 2 hydrogens, and therefore becomes NADH. NADH carries electrons to the transport chain, and then, as I mentioned, the electron transport chain eventually gives it down to oxygen, the final electron acceptor. And when you combine those with oxygen, remembering that electrons are really kind of falling along with hydrogens, it makes water and can release energy at the same time. So here's just showing you that is a particular shuttle molecule. You have NAD over here. It's currently oxidized, meaning that it lost its electrons, so it's positively charged, because electrons themselves are negative. So when you lose an electron, you become more positive. And then that combined with some hydrogens from food, remembering that hydrogen as an element has protons and electrons involved, and then 2 of those electrons and a hydrogen come to make NADH, and then some of those hydrogens kind of come off over here, and that's going to create a gradient that we'll see later on to make ATP. Here's the electron transport chain, similar in concept to what I mentioned before. If on the left you just have an excited electron and then it comes way back down, it's going to explode and release energy. However, if you have an excited electron and it slowly comes down as a kind of controlled release down the electron transport chain, eventually to the final electron acceptor, oxygen, it will make water, and we can harvest ATP in that process. So overall, the stages of cellular respiration include 3 major parts: glycolysis, the Krebs cycle, and there's kind of like a little tiny baby step before that that's in between glycolysis and the Krebs cycle called pyruvate oxidation. We kind of lump it together in with the citric acid cycle. And to be clear, citric acid cycle and Krebs cycle are totally the same thing. It's called citric acid cycle because it generates citrate at a certain point, but also some dude named Krebs was the one who really discovered the cycle. So that's why it can be called both. The final piece is where the electron transport chain comes in. This is oxidative phosphorylation. There's a second type that we're going to learn about previous, but it combines electron transport chain and the end part, chemiosmosis, which is where the ATP is actually generated. This is just the overall picture of where these elements are happening. So glycolysis, for example, happens out here in the cytosol, and that's where glucose is converted into something called pyruvate. We'll talk about it. A tiny bit of ATP is made. Some shuttle molecules, NADH, are made that get to be used later on over here in the oxidative phosphorylation piece. Then you move into the mitochondria, our powerhouse of the cell, into the open space where there's a pyruvate oxidation. And then in the matrix also is where the citric acid cycle happens. Some of the ATP is made, some of the shuttle molecules, we have NADH again, and we also have FADH2. Those things come together at the very, very end in the electron transport chain to generate a lot of ATP. And that part happens in the membrane, which we will talk about. Okay, so glycolysis. First part. All these bullet points. So key pieces. Glycolysis means sugar splitting. Lysis, as we learned, means to break. So we have had hydrolysis to break with water. In this case, glycolysis literally means to break sugar splitting. It's thought to be a very ancient thing because we believed that once upon a time, in the atmosphere, there wasn't as much oxygen readily available as there is now. So organisms had to make do without oxygen. And so glycolysis, as we will learn, does not require oxygen to happen. And later we'll talk about the organisms that just do this part, because there is elements where organisms can generate energy in the absence of oxygen. Those are anaerobic respiration. This occurs in the cytosol, so outside, just in the floaty space near the mitochondria. It partially oxidizes glucose, so it chops it in half, essentially. So it takes the 6-carbon compound that is glucose and chops it into 2 3-carbon molecules. So in this element, it would be 2 pyruvate molecules. Overall, there's a net gain. So I'll talk a little bit about there is a bit of an input of energy that's necessary, but at the end, we get 2 ATPs from substrate-level phosphorylation and 2 NADHs that will carry electrons to the electron transport chain. In this process, water is also made. Okay. In glycolysis, first you have to put in a little bit of energy to make this whole thing kind of happen. And it's a little tricky because I did say that cellular respiration is an exergonic process where energy is given off and spontaneous, blah, blah, blah. But there is this tiny, tiny little bit of energy investment necessary. So it's going to end up using 2 ATP and making 4. So overall, you have a net gain of 2. So there's an energy investment part and then the payoff part where the 2 3-carbon compounds are oxidized, and then overall for each glucose molecule you get those 2 ATP and 2 NADHs. Substrate-level phosphorylation just means that there's an enzyme that takes a phosphate and shoves it right back onto an ADP. So it brings the 2 together and joins them. That's why it's called substrate-level phosphorylation, because ADP is kind of being joined to a phosphate that is acting as a substrate. So the enzyme does the work. It is able to do that transfer and shove the phosphate back on so that the ATP molecule is harnessing a little bit of energy that can be used later. So this happens in glycolysis. When we say that net gain of 2 ATP, it is done by this method. Here's the super drawn out picture. You do not need to know this, in my opinion, in any way, shape, or form in detail. This just shows you kind of at the top to go from glucose to those 2 3-carbon molecules across the top. You have to invest 2 ATP just to make it happen. And you can see where those phosphates go. On the bottom, you can see the energy payoff phase. Kind of in that initial step, you make your 2 NADHs that will travel later to the electron transport chain. And then you make a total of 4 ATP, so over a net gain of 2. And here also in step 9, you can see that some water is given off. So when that was referenced, it's happening there. You do not need to know the super specifics. Just know that glucose goes in, 2 3-carbon molecules of pyruvate come out. There's a net gain of 2 ATP and 2 NADHs and 2 waters, which is pretty much what this is right here. So this is a good slide to just memorize as a summary. There, now it's more complete. All right, so now we're on to stage 2. So there's this weird little intermediate step called pyruvate oxidation, and then we get to the citric acid cycle, aka the Krebs cycle. So this is finally inside the mitochondria. We've been hanging out in the cytosol up to this point, but now we're actually entering the mitochondria to make more. This step is going to happen in the matrix, so that's kind of the fluid inside the membrane. If we remember, the mitochondria has an inner membrane that has those weaves back and forth to increase overall surface area, and we already know why that's important. And then there's an outer membrane. So the citric acid cycle or Krebs cycle happens in the matrix, and then finally the electron transport chain is going to happen in the inner membrane, because that's the part that's going to require a gradient across a membrane. Here is what this overall in general looks like. At the top, we have the pyruvate oxidation. So up at the top, we've got our 2 pyruvate molecules that happened to appear via glycolysis. It makes its way into this matrix of the mitochondria, and magic happens. Basically, one of the carbons is chopped off, generating carbon dioxide, and in that process, CoA is going to take its place, coenzyme A, to generate a molecule called acetyl-CoA. And in that overall process also, NAD gets converted to NADH. So another one of those electron carrier molecules is generated. And this happens twice because there's 2 pyruvate molecules. So you get 2 NADHs, 2 acetyl-CoAs that are going to be generated to go into the citric acid cycle. And we'll talk about that in more depth. Pretty much what I just said. Pyruvate is, in this particular step, generated into acetyl-CoA. So CoA is a molecule that kind of pops in for this point in time. It gets recycled. It'll eventually leave and kind of do the same thing over and over and over again. But acetyl-CoA is what we use to make citrate, which is why it's again called the citric acid cycle. Carbon dioxide is given off because we chop off a carbon to make this happen. And an NADH, in this case 2 of them, 1 for each pyruvate molecule, is made. Which brings us to the citric acid cycle. It occurs in the matrix, as I mentioned. Oops, too far. Acetyl-CoA gets converted into citrate, and carbon dioxide is released. I will show you the circle in a minute to kind of make it clearer. Overall, in this process, we are going to make 2 ATP. This is, again, just like glycolysis, going to make via substrate-level phosphorylation. We will generate 6 total NADHs, and 2 total FADH2s. FADH2 is also an electron carrier molecule, so it's going to carry to the electron transport chain also. It just doesn't carry as much. And then ATP gets produced by that substrate-level phosphorylation. Here's the super in-depth version. You do not need to know every single step. Although, believe you me, I did have to memorize all of these multiple times in college. Hopefully that's not a thing anymore. But at the very top, you can see acetyl-CoA is coming in, and then CoA will eventually get kicked off and recycled. But this will work its way through. Citrate is kind of the first piece of what gets made in the citric acid cycle. Water kind of comes in and out in this process. So you can see over in step 7 also, there is an input of water that needs to happen. Throughout the cycle, you can see there's 1, 2, 3 NADHs made in step 3, 4, and 8. Keeping in mind that there are 2 acetyl-CoA molecules that enter. So for every glucose that we start with, there are 2 rotations of the citric acid cycle. So there's 3 generated per cycle, which gives us a net total of 6. Same thing with FADH2, that will then create 2 total. And ATP down here at the bottom, which is generated via substrate-level phosphorylation, we will end up with 2. It is important to note that NADH has a little bit more in terms of how much energy it can carry compared to FADH2. And it has to do with where they drop them off at the electron transport chain, and I'll get to that in a little bit. Okay, so summary of these 2 things. We had 2 pyruvates that in that pre-step got converted to 2 acetyl-CoAs. And then they went through the cycle, and then we kind of ended up with 2 of these guys. If I go back to the previous step, that's what we end with, oxaloacetate. There's a net gain of 2 ATP by substrate-level phosphorylation, a total of 8 NADHs, 2 from the pyruvate oxidation pre-step, and 6 from the citric acid cycle itself. We get 6 carbon dioxides total. You can see from the citric acid cycle, we had 2 times 2 turns, so 4 total from the citric acid cycle and 2 from the pre-step. And then we had 2 from the citric acid cycle for FADH2. Which collectively comes to the 3rd step, the other type of phosphorylation, oxidative phosphorylation. Kind of highlighting the 2 here, how this is happening. It's a combination of the electron transport chain, which we did learn a little bit about in photosynthesis. This is occurring in the inner membrane of the mitochondria. When all is said and done, we will have 26 to 28 ATP made in this step using chemiosmosis, which is explained on the right. What happens here is we create a proton gradient, essentially, in the membrane. So what happens is we pump all these protons that have been carried, or electrons essentially, that have been carried by NADH and FADH2, and they pump through the electron transport chain. As electrons are falling down, the protons are pumped against their concentration gradient, so that kind of creates an even stronger gradient. And then they will diffuse through an enzyme called ATP Synthase, and that fuels the necessary work to generate ATP from ADP. So we'll talk about this in depth, but here's kind of what the electron transport chain looks like. So it's a series of a whole bunch of molecules embedded in the inner membrane. There are protein and non-protein components to this, not really super important, but these things basically alternate between reducing and oxidizing the electrons, and then it slowly hops down the train. And in that process, it will go to its final electron acceptor, because it's super electronegative, oxygen. and then that oxygen will combine with some of the protons, and that will generate water. So another thing that kind of creates that gradient is the H's that this thing takes away by creating water. It helps make a stronger deficit that will be used later. What you will also see is that some of the energy, this is where NADH is dropping off its electrons up here at a higher level in the chain, and FADH2 is actually dropping it off at a slightly lower level in the chain. So what eventually happens is that NADH, by where it drops it off, every one of those can generate about 3 ATP, whereas FADH2s can only generate about 2 ATP. So overall, the electron transport chain itself does not make ATP directly. It is passing down electrons to create this big gradient that will be used later. And here's kind of what that looks like. So as electrons move through the electron transport chain, then protons are being pumped into the intermembrane space. So you can see the matrix here is where the Krebs cycle took place, and then on the other side is the intermembrane space, and you have the inner membrane here. So we're pumping all of these protons onto the one side, so there's a heavy concentration gradient. And through that process, you can see NADH is dropping it off a little upstream, FADH2 drops it off at a little downstream. You shouldn't have to know the specific complexes involved. At the end, we've got oxygen here as our final electron acceptor, and it combines with some protons to again make water. And you can see how if we have a whole bunch of protons at the top, and then we're also removing some from the bottom, that just makes the gradient even larger. Which brings us to the chemiosmosis part. We've learned that osmosis really is like the movement of water. In this case, chemiosmosis is the movement of a chemical. It's moving down its concentration gradient, but we can use the energy that it's using to do this to make ATP. It's like a little pump or a rotor, so to speak. You can see a little bit in the picture. Chemiosmosis is the proton gradient that drives the work. It's the same thing as proton motive force, if you ever see that. ATP synthase is the overall enzyme that is making this happen. You can see its complex structure. But it uses energy from the proton gradient flow back across the membrane. Here's what it looks like when you put it all together. And this is oxidative phosphorylation because it has to do with the oxidation of these molecules. So NADH drops off its electrons and it becomes oxidized. It's losing electrons. FADH drops off its electrons. It's losing electrons. So this whole thing is kind of the oxidation piece, and then that oxidation fuels the proton gradient that can then make ATP. Okay, so oxidative phosphorylation uses chemiosmosis to generate ATP using redox reactions of the electron transport chain, in which electrons are passed down several energy levels to oxygen that makes water. Got a little cut off there at the bottom. Chemiosmosis couples a proton gradient called a proton motive force to drive protons through the ATP synthase, which then that process can produce ATP. And then in the process of electrons getting passed down the electron transport chain, protons are pumped from the matrix to the space to generate the proton gradient. So if this graphic is helpful, hopefully it kind of puts it all into perspective. Here you can see where everything is being made. So in glycolysis, we have a net gain of 2 ATP. Remember, we had to put in 2 to make 4, so net gain of 2. We also made 2 NADHs that will then act as a shuttle later on. There's the little intermediate step of pyruvate oxidation that generates 2 NADHs. that can be used later on in the oxidative phosphorylation part. 2 more ATP is made in the citric acid cycle by substrate-level phosphorylation, and then these little intermediate carriers all shuttle over to oxidative phosphorylation occurring in the inner membrane, and that causes the proton gradient to feed through ATP synthase, generating about 26 to 28 ATP. So overall, per every glucose molecule that is made, you can make about 30 to 32 ATP molecules. Okay, so now we're going to shift gears a little bit. What happens when there isn't oxygen? What things can still happen? So this is where, up to the point, we've been really talking about aerobic respiration. Aerobic respiration means that it requires oxygen. And in this case, you can't go through the Krebs cycle without an input of oxygen. So here we have anaerobic respiration, which is able to make ATP using something that doesn't have oxygen as an electron acceptor. So the final electron acceptor could be sulfate or nitrate or sulfur, something else. And as we'll see, it generates ATP, but not nearly as much as it could have otherwise. There are organisms called obligate anaerobes, which literally cannot survive. They are obligated to be anaerobic. They cannot survive in a world that has oxygen. And there are also something called facultative anaerobes. These are the kind of equal opportunity guys. They can make ATP by aerobic respiration when it's there, or they have the ability to switch to fermentation if they need to when there's no oxygen available. So our muscle cells are an excellent example of this. In a perfect world, they are using oxygen to make ATP via anaerobic respiration because it makes more, but upon strenuous exercise, maybe those muscle cells used up all the necessary oxygen. They don't have any more, so they switch to fermentation. And that's when we get something called lactic acid buildup, and that's why your muscles hurt after you've done strenuous exercise. So fermentation itself is really glycolysis to generate pyruvate, and then regeneration of NAD+. So we talked about the reduction of NAD+ to NADH, and then it kind of regenerates that process. After glycolysis, if oxygen is present, then magic happens. We go into the mitochondria and do everything that we've previously talked about. However, if oxygen is not present, then the cell does something called fermentation. And this is where things like ethanol or lactate or lactic acid, as I mentioned before, other products are made. So fermentation really is the process of respiration without oxygen. Here's what they look like side by side. Fermentation keeps glycolysis going by regenerating NAD+. So by regenerating this molecule, you can do glycolysis again. Whereas in respiration, it's really from the breakdown of food in the presence of oxygen. They both occur, in this case, glycolysis. Afterward, fermentation occurs in the cytosol, whereas respiration then takes place in the mitochondria. Fermentation does not require oxygen. Respiration does because oxygen is the final electron acceptor. Fermentation can generate ethanol and carbon dioxide or lactate, lactic acid, whereas respiration does not produce those things. Instead, it produces carbon dioxide and water and up to 32 ATP, where fermentation only does 2, and that's really from glycolysis, which is the same 2 that's generated in the respiration side. Okay, so there's 2 types of fermentation. There's alcohol fermentation, which takes pyruvate and converts it to ethanol and CO2. So I always think of bacteria or yeast. Baker's yeast is an example of this. It's also used in winemaking, obviously, brewing of various sorts, because ethyl alcohol is where that comes from. And it also can generate carbon dioxide. Lactic acid produces lactate. So fungi, bacteria, our muscle cells, we use this to make a lot of cheeses and yogurts, as well as methanol and acetone, so nail polish remover. Sounds really crazy. Lactic acid buildup, as it's said here, is thought to be the main cause of fatigue and pain, as I mentioned previously, but it's kind of an older idea that has been debunked a little bit. Various sources of fuel. So where do we get the food that we need to break down the things that we need in order to generate energy? So we've talked mainly about glucose, right? Like glucose is a carbohydrate. It is a monosaccharide sugar carbohydrate, bringing in all of those vocabulary words, and then it goes through this process. But fats and proteins can all be used as fuel for cellular respiration. So monomers enter either glycolysis or the citric acid cycle at different points. Carbohydrates enter in glycolysis. The sugars enter in that step. Fats can either enter in the glycerol, somewhere in the middle of glycolysis. They can also enter, kind of bypassing glycolysis in the acetyl-CoA phase. Proteins are also enter a little bit later on. They skip over the glycolysis. And that's why, depending on what is happening, you make different amounts slightly of ATP. This last slide here relates to feedback inhibition. So at the end of this overall process, something called phosphofructokinase, we have an enzyme here, is inhibited by ATP and citrate also. But the idea here is that if you've made enough ATP, let's say your cell is fully charged, ready to go, or you have a lot of citrate, those things do feedback inhibition. Their presence in high quantities inhibits phosphofructokinase so that overall glycolysis cannot fully take place and you cannot convert to pyruvate. And that will prevent the further action of happening through respiration. And then over time, ATP will go down and citrate will go down, and then that will release the inhibition. AMP is adenosine monophosphate. That's ATP that has had 2 phosphate chopped off. So if you ever have that, that means that you're really, really low on energy because you've chopped off the phosphates to release energy with that. So then it would stimulate, meaning, oh my God, cell, you need to make way more ATP. Okay, so big picture. Putting it all together, I am just making it all show. Energy, if you have oxygen, then the left side's basically what's going to happen. If you don't have oxygen, then the right side's going to happen. Here's glycolysis in the citric acid cycle. You know, big picture review, overall inputs and outputs. Oxidative phosphorylation is, again, electron transport chain with chemiosmosis to generate most of the ATP. So these last couple slides are a really good big picture view of kind of what's going on. And that's it. Piece of cake, right?